Now, previously we did about club moss, which was Lycopodium. So today let's look at spike moss, okay, which is Selaginella. So here is the systematic position. You don't really need to know because you've learned the classification, no? Okay, now let's look where we can find it. There are about 700 species of Selaginella, okay? We commonly call this as spike moss or small club moss, okay? So it's spike moss. We find them in the warm places like our place. So tropical to subtropical in distribution. Some we find them in the temperate region also. So in India, as I said, we are in the hot spot, no? So we are in the eastern Himalayas. We always find most of the plant species. So in India, we find them in the eastern and western Himalayas and in the hilly regions of South India. Okay. So we have about 70 species in India. Okay. Let me say again. Worldwide, there are around 700 species. In India, we have 70 species. So they are found in the warm, that is tropical and subtropical. Some are there also in the temperate regions. And we find in India, western, eastern Himalayas and South India hills. Okay. So the common Indian species we find Selaginella, Megaphylla. You can remember just a few, enough. Megaphylla. Then you can remember rupestris, okay, Selaginella rupestris. Then this one from the word bryophytes, no? So bryopteris, because they look a bit like bryophytes, a little bit like ferns. Ferns, the word is pteris, no? So bryopteris, okay, in between bryophytes, in between ferns. So you can remember at least one or two will do, okay. Then let's look at how it looks like and where it likes to live. So the habit is how it looks like. It shows different sizes, different shapes, and different looks. Okay. Some are like herbs and they live throughout the year, that is perennial. Few are, they die in 1 year, they live and die in 1 year, so annual. The annual one is Selaginella pygmaea, okay, from the word Pygmalion. Pygmalion is a play, no? Remember? Eliza Doolittle. I don't know if you remember or you know this play. Okay. So you can just Google. It's a very beautiful play, okay. So majority are flat. Do you remember? We had seen, no? Selaginella and Lycopodium. We saw that the Selaginella was flat, right? It had those flat leaves, while Lycopodium had the round stem. So, since it is flat, so most of the Selaginella are dorsiventral, okay? They are flat, okay? They are prostrate, means they are prostrate and creeping like grass, all right, on the surface. So, the example is Selaginella crossyana, okay? The one that you have drawn. No picture or what? Okay, here. You can see this one. No, it is flat, prostrate. You can see the two types of leaves. It has a root known as a rhizophore. Root known as a rhizophore. It has some leaves, big leaves. There you can see. You can see small leaves. Those are the small ones. And then you have some big leaves. All right. Let us continue. So there are other types also. Some are radial, means they are rounded and they grow straight up, so erect. Some are climbers, all right. Then most of them, they grow in the ground in the wet places, the humid places and the shady places, okay. You will always find, okay, these ferns, in the moist place, okay, even in your house, because we are in the eastern Himalayas, no? In your house or in any place where you find some soil which is humid, which is shady, no, you will find some ferns, okay. All right. So some species, they like to live on the dry surface, that is xerophytic. So they grow on the exposed rock surface. Example is Selaginella lepidophyta, and they can survive very dry conditions. Then there is a use of these plants, these xerophytic plants. Once they are dry, no, you can store them for many years, okay. They will roll into a ball and it look like it's just a layer of dead leaves. You will just look like a ball of dead leaves, but as soon as you throw it in water, it will live again, you know. So like Jesus, no, he was resurrected on the third day. So these plants are also known as resurrection plants. You can see them, obviously not here because we'll find them only in the desert. You can see them, some dry ferns. They will be curled up and they will survive for many months, okay, in the dry condition. Once the water comes, maybe through the rains, again they will open up and they will carry on their normal activity like as if nothing happened, okay. So such plants, we call them as the resurrection plants. All right. Now let's look at some features of Selaginella. It is herbaceous. It's like a herb, no? Then the shoot is flat. Remember, it is dorsiventral. The leaves are small and a ligule is present at the base, okay, of each leaf. So you remember I have drawn you that ligule. Then rhizophore is a structure, no, from where the roots will arise, okay. There are no leaves that appear from the rhizophore. So we will look in detail, okay, about the rhizophore later. So we will have sporophyll. This is where the sporangia will appear. Okay, now let's look at the plant body. Okay, like I told you, we can differentiate them into root, stem and leaves, and they also have this structure from which the root will appear, that is the rhizophore. Okay, so just looking at this image, you can describe from there, no, that it has a structure known as a rhizosphere, which doesn't have any leaves, and from the rhizosphere will appear the roots. Okay, then you can say that there are two types of leaves: small leaves and large leaf, microphylls and megaphylls. Yes. Then you can say that there's a ligule present at the base of each leaf. All right. Now let's look at the stem. So I've already drawn, no, the stem. Remember, it has outer cuticle, epidermis, then it had a hypodermis, then cortex, then it has those trabeculae, those swing. Remember the swing? And then a core of phloem, and in the middle you have the xylem. Okay, so from here itself you can describe about your stem. Let's just look at what they have written. I'm sure you have all, even without me teaching, you can all write about the stem. You can draw it also, because I could see that you caught on very fast that day. Still, we will just see, okay? Now, there are two types of Selaginella, okay, subtypes. One is homeophyllum and one is heterophyllum, okay? The one that stand up straight is the homeophyllum. The one that crawl on the floor is known as heterophyllum, okay? The branching is, like I said, like a V, dichotomous. Now, we cut the TS, we see that they have an outer epidermis, a cortex, and then they have a cuticle also. There are no stomata. Then they have hypodermis. The cortex is made up of compactly arranged parenchyma cells. Then most part of the cortex in the dry species have thick walls, okay, so that they will retain water. Then the number of stelae varies from species to species and even in the same species, okay. So the one that we shown, this one, the stelae, it can be more than one, okay. The one that we shown, it has only one. Okay. So the one that is creeping on the floor, the rhizome has only one stele. It is protostelic. All right. So it means they have a single cylindrical protostele. Okay. Whereas the flat one may have two or more vascular strands. So let's. Okay. Suppose this one is the epidermis, so and so. This is the space. Sometimes you can find two. Okay. Two people are swinging like this. Okay. So here you can find two types of stele. All right. So each stele, they are surrounded by a pericycle. You remember, no, from the drawing. And the stele comes off from the cortex by a few elongated endodermal cells designated as trabeculae. Okay. These trabeculae, right, they have Casparian bands on their radial walls. Okay. Let's say that this is the trabeculae, no? Let's say this one, and that's the stele. Here, there is Casparian band like this. A band of Casparian. Okay, these are a type of cells that help in conduction of water and minerals. So the xylem is the xylem is exarch. Means the old one is the metaxylem and the young one is the protoxylem. Then the phloem consists of sieve tubes, sieve cells and phloem parenchyma. All right. We won't look at this diagram yet. In some species, the xylem possesses true vessels with transverse perforation plates. Okay. So xylem, it has tracheids, it has xylem vessels. Okay, so there are different types of xylem cells, different types of phloem cells. The subtypes of phloem cells are phloem parenchyma, sieve tube, sieve cells. Okay. So even a xylem, they have different subxylem. Okay, they are some are tracheids, some are vessels. Okay, you can just Google the types of xylem, the types of phloem. Okay, so here the scientist is just telling you about the type of xylem and phloem that you find. Okay, now here look at this diagram of the root-like structure known as the rhizophore of Selaginella. So again, as I told you, we always will start with the diagram. Once you know the diagram, you can explain in your words. Okay, see, so you have a single layer of cells known as the epidermal cells. Then below that you have some thickened cells that are known as sclerenchyma. Then we have the cortex. 1 minute, I will just scroll. You have the cortex. Then it's a very big cortex. Then deep inside the cortex, we are finding endodermis layer. Then we find protoxylem and surrounding the protoxylem and metaxylem is the cylinder of phloem. So very easy structure. So let's look at what they've written. So rhizophore come from the Greek word rhiza means root, phora means bearer. Okay. So in many species of Selaginella, there is a strange cylinder-like structure which originate from the stem at the point of the branching. Okay, from where the branch will originate, some structure come out, and then they will grow downwards, and from this structure, numerous adventitious roots will appear. So these are known as the rhizophore. So a TS of the rhizophore will show very much similar to the root. Okay. However, there are few differences. Here we see that the epidermis is just one layer and it has a cuticle, a cutin. Okay, cutinize. Okay, it has a cuticle. I did not see any cuticle in the diagram. Maybe we have to draw by ourselves. Okay. So the cortex, we saw it was such a long cortex, no, wasn't it? So it's extensive and it may be differentiated into an outer thin-walled parenchyma and an outer thick-walled sclerenchyma zone. So you remember those thick thick cells, those are the sclerenchymal cells, and the down ones are the parenchymal cells. Okay. The stele again is protostelic, the most common one, no? The most primitive one, which is central xylem surrounded by a cylinder of phloem. Okay. In Selaginella martensi, the xylem is monarch and exarch. Okay, these are the arrangement of the xylem. Then here some you will find they are monarch, some are centroxylic. The explanation is given there. Okay, what is centroxylic, what is monarch, it's given. Okay, these are just the way that the xylem and phloem are arranged. Now the morphological nature of the rhizophore, it is, the scientists are still fighting about it. Okay, they are still arguing because of the unusual position. Okay, because it came out from the branch. Some people are saying it will be a stem. Some people say no, just because it came from the branch doesn't mean that it is a stem, because it behaved like a root. How will I say? Okay, suppose you have one extra arm coming out from your hips. So is that an arm or is that a foot? We don't know, right? So we will check. Okay, we try to see some similarities. Are there any similarities with an arm or with a foot? So here, how is it similar with the root? Okay, it's similar with the root because they grow towards the soil. So they are positively geotrophic in nature. Okay, similar with the root because they do not have leaves. Then the way the xylem is, the central xylem is surrounded by phloem. It looks like roots. Mostly roots have that xylem phloem arrangement. Then there are root cap in some species. Okay, like Crocyanea, they have a root cap. Then transport of auxin in rhizophore is acropetal, which is similar to the root. Okay, it goes towards the tip. Now, how it is similar with the stem? Okay, it is similar with the stem because it is exogenous in origin, like the stem. Means it just came from one branch, exogenous. Okay. Then in most species, they don't have root cap and root hairs. Okay. In roots, you should have root cap and root hairs, but here only few species. Like Crocyanea, it had root cap, but most of them, majority don't have root cap, and root hair is not even there. Okay. Then the way they appear also is because of the activity of the meristem. Now, to explain to you what is a meristem, try to imagine yourself as a baby. Okay, you came from embryonic cells. Okay, you came from embryonic cells. At one point of time, you were just a zygote, and that zygote divided into a group of 16 cells, which are highly meristematic embryonic cells. Okay, all those 16 cells have the capacity to form a brain, an arm, a leg, a foot, intestine, etc. All those 16 cells, which means that out of the 16 cells, if one cell is damaged, 15 are still there, to form the head, the arm, the root, the shoot, whatever. So these are meristematic cells. Okay, so let's say you want to clone yourself, okay, or your mother wanted to have a twin or triplet. Okay, so she will take out those embryonic cells, okay, which are highly meristematic, take one of them. Okay, there were 16, she took out one. All right, place it separately in another womb. It will develop into a complete human. It will reproduce. It will divide from that one cell. Again, it will become 16 cells. You get it? So that these cells, which have all the information to form a complete human being, These are your meristematic cells, okay? So even in plants, we have meristematic cells. These are there at the tip of the shoot, okay? Like when a plant was young, no, it was very short. You touch the shoot part. That one has the meristematic cell, okay? It keeps growing from the tip, okay? Let me just draw. Like, okay, here, okay, this is the small plant, all right? Here, this is where the meristematic cells are there. Okay, so it is not that this down part will grow. It's just this part will keep on growing. Okay, it grows. Then the meristematic cell was here. Again, this part will not grow. This part only will grow. So it grew like that. So these are your meristematic cells. Okay. So because the root, this rhizospore, originated from the meristematic cells, and meristematic cells are supposed to be present in the shoot, so they are saying that it has to be stem. Then some people, they perform some experiment where that rhizospore, it converted into a leafy shoot. Okay, a leafy shoot or leafy short. So that was why they concluded that it might be a stem. So one person, Bauer and Gobel, they said it's neither root nor stem, but an organ which came by itself, that it is generated by itself, sui generis. Then another scientist says that it is a specialized stem which behaves like a root. Okay, it's just a special stem which behaves like a root. Then there are some biochemical studies where they study the protein from the leaf, the stem, the root, etc. And they saw that the proteins, okay, the proteins of the rhizospore, they were closely resembling the stem rather than the roots. Okay. So that's why we do not say directly root, we say rhizospores, okay, because they could be either root or stem.